Hello, everybody. Thank you, Francesca, for the introduction. I am very pleased today to present to you this webinar session called Metagenomics, the Secret Life of Microbes. Just before going into details in the presentation, I would like to present my team, the Centers of Excellence. We are a team working on microbiota and the relationship of the microbiota with the health of the host. And we are 6 scientists, 2 project leaders, and 1 technician. And some of the scientists are located to different research center, and so it makes a unique and original link between academic research, applied research, and economic world. And also we have a preferred access to different analytical platforms. Within the team, we have 2 scientists dedicated to amplicon sequencing and omics analysis, and you can see at the bottom their picture, Caroline Achard and Anna Rodiles. On the next slide, I would like to give you the main objective of the presentation. The first one is to explain the basic of the high throughput methods, and we will do a focus on amplicon sequencing. And the second objective is to explain to you how those techniques bring value to understanding the mode of action and benefits of microbial solution in pet and equine nutrition and health. As an introduction, I am sure that you know that we are made of microbes, and this is the same for pet and horse. And we can speak about the gut big data. Indeed, on the slide, you can see few figures on the importance of the microbes in our intestine content. And again, for dogs, cats, and horses, you can see few figures. And you can imagine that all these bacteria represent a lot of genes. And these genes, of course, will interact with the host. And you can see that there are many factors with which our gut microbiome will interact. It can be lifestyle and behavior, drugs, age, illness, stress, and diet. So you can see that this is a very huge field of research. So in Lallemand, we are driving research into the gut microbiomes of cats, dogs, and horses. And the first aim we have is to bring some knowledge to find new biomarkers associated with health status and to better measure pets' health and to predict, when possible, future health. So to measure the risk of disease based on some microbiota data. And of course, the second aim for us is to be able to use our microbial solution in the best way in your products to answer specific needs of companion animals. The first one is to understand the mode of action of our solution in a specific context. And the second aim is to provide personalized solution to optimize pets' health. And as you know, microbiota becomes more and more important. And in this slide, I took an example coming from the website of the International Scientific Association for Pro and Prebiotics. And you can see that this is a page dedicated to the consumer, so for available for everybody. And you can see that they speak about probiotics, prebiotics, and even global biotics, because microbiota and the study of the different microbial communities becomes very important. And this big explosion of research is mainly due to the big change in science, thanks to the apparition and the evolution of molecular biology and high-throughput sequencing that have changed our life of scientists. And in this slide, you can see the exponential increasing of the number of publications using the word microbiota since 2007 to 2017. You can see the number of publications really increase. And this molecular biology and high throughput sequencing method are in fact some tools that allow us to better analyze composition, activity, and interaction of the microbiome. and to decipher the response of the host. But from time to time, this method can seem complicated, and what we would like to do today for you is to give some tips to make the understanding easier. Indeed, in this slide, you have a kind of landscape of the different methods we have that are available to work on the microbiome topic. And you can see that we can go from the microbial potential to the microbial function on the right. And for that, we can apply different types of methods. First, we can start by the study of the cells using microscopy or culture and culturomics. But we will not go into details today on that. But we will really focus on the DNA, on the study of the DNA, and we can use two main methods. First is the amplicon sequencing, and we will have the community composition, and we can do after that microbial networks, and we will go into details. And the second one is the metagenomics, for which we can analyze the whole metagenome of the microbes. And so we will have an idea of the genes, the genome, and the gene function. We can also study the metabolic potential using the RNA and doing metatranscriptomics to have an idea of the gene expression. And then at a more phenotypic level, we can study the microbial function using protein, and the method is called metaproteomics, or metabolites, and we can do metabolomics to have the metabolite production and microbial product. And so today we will focus on the amplicon sequencing. What is amplicon sequencing and just also metagenomics method? So they are analysis of the composition of a microbial community based on its DNA sequences. This is really a focus on the DNA to understand the composition. And how? We have a series of innovative methods using bioinformatics, and it is called the high throughput sequencing, or HTS. What we will have with these techniques is an identification or a quantification of the microorganism within a community based on their genetic content. And we will also describe the relationship of these microbes and the evolution according to different factors like lifespan or other external factors like diets or drugs. And third, we will understand and predict the action of the various types of molecules and metabolites produced. At the end, what do the word sequencing mean? It's the determination of the exact order of the base pair in a DNA segment. The difference between amplicon sequencing and metagenomic is that with amplicon sequencing, we will focus on one specific DNA sequence, whereas with metagenomics, we will sequence the whole genome of the microbiota. If we go a little bit into details on the amplicon sequencing, you can see in the different papers two main names, the 16S RNA or the amplicon sequencing. Why do we use this name? So first, as I mentioned previously, it is an analysis of the microbial community based on DNA sequencing. It is not necessary to sequence the full metagenome because it costs a lot of time and it is very expensive. Target only a specific gene markers, which is used as an ID card, and so it is a targeted approach. And the gene marker we usually use for bacteria is a 16S RNA. Why? Because it is ubiquitous. It combines conserved and hypervariable regions, and this is what you can see on the figures on the left. You can see that here you have the percentage of sequence identity here, and you see that certain regions, the gray one, are very, very variable, while the orange one are very conserved. And this is very useful to use your primers, and you will use the primers of different region in the gene. And you will start by the conserved region, and after that, the hypervariable region will be sequenced. And what it is interesting is that the hypervariable region vary proportionally to the genetic distance. And in addition, there is large database available to make the correct microbial affiliation. The second name we can see in the publication can be amplicon sequencing. And amplicon sequencing is used because, in fact, you will see on the next slide, we will use a amplification step using PCR, polymerase chain reaction, to create numerous copies, and these DNA copies are called amplicon. And you have here figures summarizing the process of the PCR. You will use some specific primers of the targeting region. So, for example, between the V1 and the V3, and you will put a specific enzyme, the Taq polymerase, and some free nucleotides, and you will copy the target gene in numerous different copies. And this is the amplicon. In these figures, you have the whole process of the amplicon sequencing, and you will see at the end, it's quite easy. The first step is the sample collection, so the intestinal or fecal content. And this step is quite important to be sure that the sample you will get will reflect what you have at the beginning, especially the way to do the collection, but also the storage condition are of importance. After that, once you have your sample, you will have to do the DNA extraction. And to do that, we will use enzymatic commercial kits and physical methods to lyse the cells and to get the DNA. And as I mentioned previously, the step after is to do the amplification. So you will have your library of different amplicons, and after all, you will have the sequencing. And what you will get at the end, it's around 30,000 sequence per sample. So you can imagine that once you get that, you need to do a strong or huge bioinformatics work. And this is the step after, sorry. And you can see here the bioinformatic analysis, and the first step is to do a quality check, and you will remove the bad sequences, you will remove the artifacts, and so on. This is the first step. And after that, you will get your final sequence here. And what we will do will be what we call the binning. And binning, in fact, aim at clustering the different sequence when they are very similar. And in general, we consider the very similar when the sequence are the same at a level of 97, 99%. So you can see here for the OTU1, you have 1, 2, 3, 4 similar sequence. And the same for OTU2, OTU3, and so on. And once you get this clustering, you will have the affiliation step, and then you will see, and we will use, in fact, different available database to do this affiliation. And you can see for OTU1 a correspondence with Lactobacillus, and it will be the same for each OTU. And at the end, each sample will have the precise composition and will be classified in the taxonomy. And you can see that we can go from the domain, for sure, bacteria in this case, but also the phylum and the class, order, family, genus, and from time to time, species. So once you did this process, you want to analyze the results. And the question is, what do the results mean? In fact, the biostatistic step will allow you to have three main indicators. The first one is the percentage, the relative abundance. It is the proportion of a given microbial species in a microbiota sample. The second one is the alpha diversity. It is a diversity within one microbiota sample, and it is related to the richness and behaviorness. And the third one is the beta diversity, which is the similarity or dissimilarity level between different microbiota samples. And here, the purpose is really to describe the microbial composition evolution according to environmental or physiological changes, for example. So if we go into details in for the first factor, the relative abundance, it is a semi-quantitative analysis, and it is defined as a percentage of a given organism of a particular kind relative to the total number of organisms in the area. And on the right, you can see a conceptual example, and you have three samples, sample 1, 2, and 3. And you can see that in the first one, the light blue and the blue are in the same relative proportion, and that the green bacteria are less abundant. And this is typically what you see in the sample 1 in the table, with bacteria 1 14%, the other bacteria 43, and 43 for the last one. And you will get this relative abundance table. And you can have after that some graphical representation, and I put here a practical example coming from a paper which aimed to study the fecal microbiota of healthy donor horse and geriatric horses that will receive fecal transplantation to treat diarrhea and colitis. And I just put that example because you can see that you have the relative abundance for each category of horses. So the old one but healthy, the young one healthy, the young donor, so the horses selected to be the donor, and the old horses exhibited colitis. And what is interesting here is that you can see that in old horses with colitis, you have an increase in the proportion of proteobacteria, while you had a decrease of fibrobacteres. And fibrobacteres is a fibrolytic bacteria, while proteobacteria, the increase in proteobacteria is largely described to be related to what we call dysbiosis, dysbiosis being an imbalance in the microbiota. So you can see that just by exploring the relative abundance of the different phyla, we can make some link with the health of the animals. Second indicator is the alpha diversity, also called the Shannon index, and you will see that we have also other index. But the diversity, the alpha diversity allows us to have the diversity within a community or a sample. In a sample, sorry. So the Shannon index is one of the most used criteria to describe alpha diversity. It takes into account the number of different microbial species, what we call the richness, but also their relative abundance or predominance in the sample. We have also some different indicators that will take into account only the richness. For example, the number of OTUs is one of these indicators. Or we also have some indicators to take into account the evenness, so the dominance of one or several species. And in fact, they are very useful when we use all these indicators in combination. On the right, you have again a conceptual example of the Shannon index. You can see that in the sample 1 and in the sample 2, you have 3 types of bacteria. So you put a score of richness of 3. But in the first case, you see that you have much more blue bacteria than light blue and green bacteria, while in this sample 2, they are much more balanced in terms of relative abundance. So at the end, as the Shannon index take into account both richness and evenness, you can see that the sample 1 had the lower Shannon index than the sample 2. And you have to remind that the higher the Shannon index is, the more diverse and richer the microbiota is. Again, I put an example coming from a recent publication, and here the aim was really to study the microbiota of 3 types of cats. The first being lean cats, the second one being the diabetic cats, and the last one was obese cats. And what you can see here is that the author highlights a lower number of OTUs on the left. This is the alpha diversity measured at the richness using the number of OTU observed. You can see that it decreased compared to the lean and obese cats. And on the right, you have the Shannon index. And again, you can see that this index decreased in diabetic cats compared to the lean and obese one. So again, it is really relationships between metabolic disorders and microbiota. The last that I wanted to present to you today is the beta diversity. The beta diversity allows us to measure the similarity between 2 samples in terms of composition. And again, you have here a conceptual example. On the left, you have the group 1, and you can see that the microbiota composition from one sample to another one is very similar among these samples. So it means that you have a low beta diversity. So at the end, the structure of the community is very similar. But in the second group, you can see that this is much more diverse from one sample to another one. The structure of the community is much more different. So you will have a high beta diversity. And you can imagine that when you have a lot of samples and a lot of OTUs, you cannot do that just like that. And you need strong bioinformatics to use some statistical method, and this is what we call the ordination technique. And we will apply a set of multiple variables to distinguish sample from each other. And at the end, we will use a kind of again, statistical methods to be able to have a representation in 2 or 3 dimensions of all these data. And if we have a short distance between each sample, it means that we will have similar microbiota composition from one individual to another one. And if we have a long distance, it means that we have very different microbiota composition. And here I put an example of a paper. We want to characterize the nasal and oral microbiota of detection dogs. And what you can see here is that the red point, which are the points related to the oral microbiota, were clustering together, while the blue one related to the nasal microbiota cluster together as well. So it means that the structure of the nasal microbiota was significantly different to the structure of the oral microbiota. So now I want to show you some preliminary data we obtained with a study on horse. You know that anti-helminthic drug is supposed to increase the risk of colitis in the bibliography, and one of the explanations is maybe the gut microbiota modulation related to this drug. And so what we wanted to do was to understand the impact of the drug on microbiota, and this is ongoing, and I'm very pleased to present to you some preliminary results. And the second step, of course, will be to evaluate the potential of our microbial solutions in this context. So the design we used was a very easy one. We used 8 horses from now, following a longitudinal study to see the evolution of the fecal microbiota before and after warming application. And what we did was to collect fecal sample just before warming. After that, the warming was made, and we used praziquantel and ivermectin molecules. And we also collected fecal samples 24 and 72 hours after warming. We had 5 mares and 3 geldings, aged from 5 to 30 years old. And as you see at the beginning, we can choose different regions to be target for the amplicon sequencing. We target V3, V4 because there are several papers mentioning this choice. So here we will have some the alpha diversity indicators. On the left, you have the number of observed OTUs, so the richness. And on the right, we have the Shannon index. And just to remind you, the Shannon index takes into account both the richness and the eveness. And you can see that overall, we do not see a strong change before here at D0 and after D1 and D3 warming application. So the first and preliminary conclusion is that the ingestion of praziquantel and ivermectin, in our case, does not significantly modulate the alpha diversity. So it means that the diversity within a sample was not changed. The second result we had was related to the beta diversity. So if you remember, if you remind, this kind of representation allow to have a global composition of one sample. So each point is one horse at one time. So in red, you have D0 before warming and D1 and D3 in green and blue. So 24 and 72 hours after warming. And you can see that globally, all the points were blended. So it meaning that it was difficult to see a specific community structure specific to the before warming moment and to the after warming moment. In fact, everything is together. So our conclusion was that in this study, the ingestion of praziquantel and ivermectin does not significantly modulate the beta diversity. And again, it means that there is no clear change in the microbial structure of the different horses. But we also look the beta diversity in a different way. So here you do not have the time point, but one point deals with one horse, and you have 3 time points, so 3 points per horse. And what we saw, in fact, was that the beta diversity, so the microbial structure, was related to the management of the horse. Indeed, you can see 3 kind of plots. The first one was related to horse or gathered horses that were managed in box. The second one gathered horses that who lived in box and pasture half and half, and the last one was for horses in pasture. So it means that the management of the horses and probably the diets seem to play a role on the structure of the fecal microbiota. And this is something we will investigate more into details in the coming months. And finally, for this example, we also had a look on the relative abundance, so the fecal microbiota composition. And here you have the first result at the phylum level. And you can see that we have 2 big phyla, Firmicutes and Bacteroidetes, which is really consistent with the bibliography. And we find other phyla like Actinobacteria or Fibrobacter or Proteobacteria. And again, it is really consistent with the bibliography. And you can see that before warming or after warming, there is no significant difference between the relative composition. But we also look on individual evolution. So You have, for example, the 3 first bars for one horse before and after warming, and this is the same for each bar plot. And what you can see is that for 2 horses, so the Quenot D0 and Bettina horses, that is the name of the horse, you can see that after warming, you have an increase in one phylum, and this phylum is Proteobacteria. And at the same time, even if the bar it is very small, we see a decrease in Fibrobacteres. And I don't know if you remember my first example with old horses suffering from colitis, but they characterize the microbiota of horse with colitis, and the characteristic was an increase in Proteobacteria while a decrease in Fibrobacteres. So you can see that probably for certain types of horses, we have a change, specific change 3 days after warming, and this change can be related to some colitis reported in the bibliography. So it means that maybe some horses are more sensitive to other one to the warming practice. So you can see through this example that 16S RNA amplicon sequencing is an easy method to get a clear picture of the microbiota at a given time. The effect of diet and management of the horse has to be investigating in addition to the effect of warming, and we have still a lot of work to do because we want to go deeper in the taxonomy levels. Here I just present the result at the phylum level, but it is not done for lower taxonomy level. We want to increase our data set and to have a look on the individual patterns. And what we can do after is a discriminant analysis to select the OTUs that can discriminate the before and after warming. This is quite a detailed analysis, and it is very informative for us. And what we want also is to decipher the importance of individual pattern and management diet. It is really a matter of personalized nutrition, and to test, of course, our microbial solution to propose to you relevant products based on scientific data and rationale. And if you remember the effect on Fibrobacteres and on Proteobacteria, we already know that our yeast are able to decrease Proteobacteria relative abundance and to increase Fibrobacteres relative abundance in ruminants and in swine. So we can already imagine that this kind of yeast can represent a big potential for this kind of application. The third example I wanted to give you today is a study that we confounded, and this study was published by 2020. The aim of the publication was to study the relation or the evolution of the gut microbiota with the age of dogs, and they used 29 companion dogs, 14 males, aged from 3 to 13 years old, and they had 11 purebreds dogs and 12 mixed breeds. And what they did was to divide the dogs into 2 groups according to the behavioral signs of aging that were registered via test, especially short-term memory test mistake. And after they did this test, they collected the fecal samples. All the dogs were fed with different foods. It is quite important for the results. And they also applied regression tree analysis because they wanted to make the link between the age and the microbiota composition and between the memory mistakes and the microbiota composition. So the first result they get is that they define a kind of core microbiota in dogs, whatever the age and whatever the result of the memory test. You can see here the first is the dog identification. The second figures is the age of the dog, and the last one was the number of memory test mistakes. you can see further that there is a big individual variability, and it is quite logical because the dogs were fed with different foods, and they were owned by pet owners, so it means that they were managed in different ways. But they find the 5 main phyla: Bacteroidetes, here in red, Firmicutes in green, Fusobacteria, Actinobacteria, and Proteobacteria. And these phyla are really the core microbiota of the dogs, whatever the age. So it was typical of the microbiota of the dog. And very interesting, they found was a link between phenotypic features and microbiota. What you can see in this slide is the result of the regression tree model. On the left, you have the result for the age, and on the right, the result for the memory test mistakes. They did not find difference when they test the beta diversity or the alpha diversity. It means no relationship between these two diversity and the age or the memory test mistakes. But what they saw is that Fusobacteria phylum was related to the age, and more precisely, Fusobacteria decreased when the dogs became more old or older, while with the memory test mistakes, Actinobacteria increased in dogs that did more mistakes. So what it is interesting here is that Fusobacteria in humans is described to increase with age, and it is contrary to what it happens in dogs. But in humans, this phylum is associated also with metabolic disorders like IBD or different dysbiosis. In dogs, it is really a phylum which is associated with the health of dogs. So it is really in agreement with a potential different role in carnivorous gastrointestinal tract compared to the human ones. And this highlights the importance to investigate the microbiota of each species of animals, because you have some similarities for sure, but also some big difference. So we really need to have some knowledge on each species we want to study. And interestingly, in dogs like in humans, more Actinobacteria is associated with memory failures. So it means that this phylum could be a good candidate to try to decipher the risk of mistake failure in the behavior of the dogs when they become older. The last example I wanted to share to you, because this is where we want to go, is really to integration between phenotypic metabolomics and amplicon sequencing data. It's an example using obese dogs, and you can see that you have plenty of relationship between phenotypic data, like a concentration of blood haptoglobin or cholesterol, with also amplicon sequencing, with this, which are variable which summarize the main features of the microbiota. And this is, in fact, it is a little bit small, but just for you to understand, each genus D1, D3, or D2 correspond to a summary of different bacteria that were very important in this kind of relationship. And the same was done for metabolomics data. And you can see that they found a lot of different metabolites that were related to other phenotypic features like fasting insulin or fasting glucose. So it's not to go into details of that, but just for you to understand the importance to put everything all together to integrate both the microbiota and the metabolomics and the phenotypic feature, to have a global picture and to be able to see what could be the important candidates in the microbiota as a biomarker or to be modulated and to find new nutritional strategies. So at the end, again, what it is important is really to have a holistic approach. So it means that to use all these tools, but it is important to keep in mind that this is to understand and to improve the welfare or the health of the animals. So the method exists to serve hypotheses and concepts. They open avenues to understand microbiomes and their implications to enhance health, performance, longevity, and well-being of companion animals. Of course, for us to design precision probiotics, microbial solution to serve personalized nutrition, and after that, to understand the mode of action of our products. And we look forward to showing you new results on the potential of microbial solution. And just to finish my presentation, I wanted to wrap up the take-home message. The first one is that method innovation allows us to study not only microbiota, but also microbiomes. Amplicon sequencing allows the study of gut microbiota composition and structure and is based on amplification method and gene marker discovery, explaining the name of the techniques. The main features you will get are number of species present, their relative numerical composition, bacterial diversity defined as alpha within a sample, and beta diversity between samples. Amplicon sequencing, thanks to dynamic evolution of bioinformatics, allows us to also understand relationships between microbes. And in the last example with integration data, you can see that the summary of OTUs were, or several OTUs were gathered together because they had the same importance for one phenotypic features. And this is the point. Integration of data from different high throughput methods will allow us to have a holistic approach to study host microbe, microbe, and microbe environment interactions. And again, to be successful, the tools available must serve hypotheses and concepts. For us, really, it is important to understand the gut microbiota patterns in different conditions. This is really required to expect the best effects of our microbial solutions in different contexts. I thank you for your participation, and I am available if you have any questions.